Okay, let's continue. Uh, in the previous video, I've shown you 4 possible situations that could occur in Lac Operon. So let's take a look at the first situation. So when both glucose and lactose are present. So this explains how the Lac Operon is transcribed only when lactose is present. But of course, this does not explain why the operon is still not transcribed when both glucose and lactose are present. Now, why does this occur? So when glucose and lactose are present, RNA polymerase can sit on the promoter site, as you can see over here, okay, even though there is no P with that, okay? But again, it is unstable and it keeps falling off. Kind of like you are unstable, okay, when you sit on something, but it is actually not keeping you stable. So what happens is that okay, it is weakly turned on. In some cases, a lot of times it's actually not even turned on. So the switch between use of glucose and lactose is activated by products of carbohydrate metabolism, okay, known as catabolite. So catabolites from glucose breakdown okay, would prevent production of cyclic AMP or CAMP, a signal molecule which we have seen earlier on okay, in the overall structure or how Lac Operon works. So absence of CAMP meaning that no enzyme production for lactose catabolism. And of course, okay, glucose itself, when it's there, the presence stops Lac Operon from being activated. So it stops lactose being used, and of course, we know that overall system still prefers okay, to use glucose over lactose. So the switch from glucose to lactose basically depends on CAM and catabolite activator protein or CAP. So when CAP binds to CAM, the CAP-CAM complex okay, later binds to specific DNA sequence upstream of the Lac Operon O and P. What happens is that okay, the RNA binding okay, will be enhanced to activate genes from the Lac Operon. So what we know is that okay, high glucose okay, would basically would be CAM level okay, would be low. So high CAM would signal lower level glucose, which is the opposite okay, of what we know. However, when glucose is present, even at low level, levels of CAM would fall. Okay? Hence, okay, activation of Lac Operon okay, would be lacking. Alright, so this is basically the diagram okay, of the explanation that I've said earlier on. You can get a lot. This is actually taken okay, from a book okay, in one of the lists in your course information. Now, the second condition situation is when glucose is present and lactose is absent. So what happens is that we still don't get any transcription going. So a prepressor protein is continuously synthesized, situated on a sequence of DNA just in front of the Lac Operon, okay, on the operator site. So the repressor protein blocks the promoter site, where the RNA polymerase settles before it starts transcription. Now, basically the second one is quite easy, like I said earlier on in the last video. So the third condition, when both glucose and lactose are absent. So this is basically not that hard to explain, okay, and we still don't get any transcription going. So if prepressor protein okay, is of course continuously synthesized, similarly situated at the sequence of DNA just in front of the Lac Operon, okay, which is on the operator side over here. So it also blocks the promoter site where the RNA polymerase settles before transcription. Now, the last one, the only one okay, that basically seems okay, to work okay, for the Lac Operon when glucose is absent and lactose is present. So a small amount of sugar, which is known as allolactose, is formed within the bacterial cell. So this fits onto the repressor protein at other active site, okay, which is at the allosteric site. Okay, so this is a representation okay, of the allolactose. So this causes conformational change of the repressor protein. It changes its shape, so it could basically no longer sit on the operator site, or basically it couldn't bind tightly. So RNA polymerase okay, can now okay, reach its promoter site, hence okay, it allows transcription of the structural genes. Simple as that. So another protein is also needed, okay, called an activator protein. So this is the one, its function is to stabilize your RNA polymerase. And it only works okay, when glucose is absent. When glucose is present, you will not get any activator protein. So in this way, E. coli usually makes enzymes to metabolize other sugars in the absence of glucose. Now, okay, this is what happens, okay? When glucose is low, low level of CAM are high and readily binds, okay, with CAP. So when it binds to CAP, it basically will bind over here, making sure that your RNA polymerase, okay, could bind securely to your promoter to start transcription of these 3 structural genes. So you get high rate of transcription and translation. So my summary, okay, of Lac Operon with the 4 situations that we have, okay, is very simple. Okay, so when we have, okay, both this is situation 1, 2, 3, and 4. Okay, for situation 1, okay, the activator protein is usually not bound to the DNA. Your repressor protein is usually lifted off the operator site, and your RNA polymerase usually keeps falling off the promoter site. So your Lac Operon, okay, produce no transcript. Second situation, there is glucose, but there is no lactose, not even a small amount of lactose. So activated protein is not bound to DNA, and we know that the it needs an activated protein. The repressor protein bound to the operator site, of course, okay, it will block, okay, the RNA from binding. Okay, so you get no transcription. Third situation, no glucose, no lactose. Activated protein, okay, because, okay, there is no glucose, okay, it will bind to DNA, okay, but the repressor protein is also bound to the operator site. What happens is that it will block, okay, the RNA polymerase, okay, from continuing with the transcription. And another clue is that because there is also no lactose, so Lac Operon would never work anyway. So the last condition, whereby when you get transcription, okay, no glucose, low, or there is lactose. So activated protein would bind to DNA. Repressor protein, okay, is lifted off the operator site. Your RNA polymerase could sit on the promoter site to start the transcription. So these are some of the common conditions, okay, but okay, just think about it, okay. What happens, okay, whereby when you have low level of glucose but high level of lactose, what will happen? Okay? It's almost similar to the situation. Okay? So my question is, okay, I'll repeat it again, okay, for you to think about, okay, and probably bring it out when we meet online next. Okay? What happens whereby when there is low glucose but high lactose? Okay, now this is also the summary of the Lac Operon in diagram. As you can see, it's actually the same as in the table just now. And if you are interested, okay, you could watch, okay, this animation or video link. I'll try to put it in the e-learning. Hopefully it still works. I think because I wasn't teaching this last semester, I think 2 semesters ago it still works. Okay, I will check it out and I put the link in the e-learning. Go and check it out. Okay, that's it for the Lac Operon.